Thanks so much, Ludwig. So, welcome to today's session on Alzheimer's, and we have the pleasure to have Professor Zetterberg, distinguished Alzheimer's person who is well known all over the world for his expertise in Alzheimer's and biomarkers. He is active at the University of Gothenburg and also at University College of London, UK. And then we also have the pleasure to have our AlzeCure CSO, Chief Scientific Officer, Johan Sandin, PhD, and also co-founder of AlzeCure. And then you have me, Martin Jönsson, who is the CEO of AlzeCure. The agenda we have for today's meeting is that we will look at Alzheimer's disease and amyloid hypothesis, where we stand today and how the knowledge around the amyloid hypothesis has developed with new evidence that has emerged. Then we will also look at Alzstatin, AlzeCure small molecule disease-modifying therapy against Alzheimer's, which is a so-called gamma-secretase modulator. And then we will also learn more about AlzeCure as a company and our pipeline. And then in the end, we will have a Q&A where we will take questions from you as an audience. So please send in questions. And if we begin to look at AlzeCure and just give a company overview and look at our pipeline, AlzeCure is focusing on Alzheimer's disease and pain, and we are spin out of AstraZeneca. So the company was created when AstraZeneca left the CNS and Alzheimer's space. And we were initially funded from the Swedish Alzheimer's Fund. So a big thanks to the Swedish Alzheimer's Fund for all the funding we have had. And today we are active at Karolinska Institute, where we also started our activities. And we have 3 project platforms with small molecule candidates with first-in-class properties. We have Alzstatin, which is an innovative, preventive, and disease-modifying treatment against Alzheimer's disease, which we will look at very much today. Then we also have NeuroRestore, which is a novel symptomatic treatment for cognitive disorders, where we are focusing on Alzheimer's disease, and where we now also have data showing that NeuroRestore is potentially neuroprotective and also disease-modifying. And then we have a platform of pain projects. And as a company, we are listed at Nasdaq First North Premier Growth Market in Sweden, which we have been since 2018. And if we look at our pipeline of small molecule program, it looks like following. We have NeuroRestore, which is with our lead candidate ACD856, which is a positive allosteric modulator, upregulating NGF and BDNF signaling, where we are focusing on Alzheimer's disease. And here we have had positive phase 1 data showing safety, tolerability, and target engagement, and where we are preparing for phase 2. Then in Alzstatin, we have 2 programs, ACD679 and 680, both gamma-secretase modulators. And here we have both programs in preclinical phase. And last year, we selected a new additional CD, ACD680. And here we are now looking at which of the programs we will go ahead and bring into phase 1. Of course, the new molecule can be beneficial with a even better patent situation. Then we have our pain platform with 2 programs, ACD440, a novel TRPV1 antagonist against neuropathic pain. And here we last year had a positive readout in a phase 2A showing proof of mechanism with safety, tolerability, and efficacy on pain. And then we also have ACD137, which is TrkNAM, a negative allosteric modulator downregulating NGF signaling. And here we about 3 weeks ago shows a CD, ACD137, which we now are preparing, working on in preclinical phase, preparing for phase 1. And what's so nice when it comes to the Alzheimer's space is that we, since the last 18 months, really have seen positive progress. Of course, we have seen scientific validation, which we will talk about today, showing what the positive benefits of reducing the amyloid load and also having positive phase 3 and approvals. And what we are now also are seeing is increased investment in this field, with additional big pharma companies entering or re-entering the field, such as Bristol Myers Squibb, MSD, and AstraZeneca. And we also see more funds investing in the field of Alzheimer's. But still, if we have this positive development, we still do not have any curative treatment or effective cognitive enhancing treatments. And this is what we at AlzeCure really would like to change. And if we look at the antibodies today, based on current published data, we know that only about 5 to 8% of the patients who come to the clinic is eligible for antibody treatments. And this really shows the huge unmet medical need that still remains with all the patients that's out there. And the way we are focusing on this from AlzeCure's point of view is that we have our 2 Alzheimer's platform. So Alzstatin, targeting amyloid production to reduce the production of the toxic amyloid A-beta 42, and then also having NeuroRestore, track PAM, positive allosteric modulator, upregulating BDNF and NGF signaling. And what's great here is that both our programs are being validated by, for instance, Roche and Eisai. Roche having a gamma-secretase modulator, and then we have Eisai having a TrkA PAM, which we will come back to. And if we begin to look at NeuroRestore, where we develop NeuroRestore to improve learning and memory capabilities, which now is in preparation for phase 2. This program, NeuroRestore, our TrkA PAMs, are focusing on the neurotrophins BDNF and NGF signaling, which we know are key in Alzheimer's. We know that these neurotrophins are key for brain health and cognition, and that when BDNF and NGF signaling are decreased, we also see negative effects with regard to cholinergic function and synaptic plasticity, and also that loss of NGF-dependent cholinergic neurons correlates with cognitive decline. And we can also see that certain genetics, like, for instance, BDNF Val66 MET polymorphism, where you see lower levels of BDNF, are leading to a more rapid cognitive impairment and increased disease progression. And that's why we are focusing on these neurotrophins, since we know that reduced BDNF or NGF levels could limit the brain's ability to withstand pathological conditions. And if we look at NeuroRestore, which is a positive allosteric modulator, which is increasing the NGF and BDNF signaling, we initially focused on developing it as a symptomatic treatment with long-term benefits. What we have seen and generated data on and published on last year is also that we see the potential also for disease-modifying effects, where we have seen that NeuroRestore is improving neural proliferation, show neuroprotection, stimulate neural outgrowth, improve synaptic plasticity as measured by SNAP-25, and also improves mitochondrial function. And the preclinical data that suggests disease-modifying effect is that we see this neural outgrowth in PC12 cells, which is with concentration similar to what is found in CSF in our MAD study. We also see increased level of synaptic markers in PC12 cells. We also see increased proliferation of TRAC receptors. We see improved mitochondrial function and neuroprotection. We also see enhanced synaptic plasticity in the hippocampus. And we have also been able to demonstrate long-term plasticity effects after repeated dosings. So this data is really supporting also the potential with the NeuroRestore as a disease modifier. And our data is also validated by external parties like Eisai. Eisai have a TrkA PAM E2511, and they have published data and also are out stating that they see this TrkA PAM as a potential disease-modifying therapy against neurodegenerative diseases in general. So this data is also helping us at AlzeCure and increasing the interest in NeuroRestore. If we compare Eisai's compound E2511, which is TrkA PAM, which is increasing NGF signaling, and then compare it to NeuroRestore ACD856, our clinical candidate, we see that there is truly a difference. We can see that both compounds is showing signs of improving or showing potential disease modification. But what we also have with our compound, which we think is due to that we also increase BDNF signaling, is that we see a cognitive enhancement, and we also see an antidepressant and long-term effects. And also this when we add it to SSRI. So this compound don't only have the opportunity in cognition, but also in depression. And if we look at ACD856, we see that it can improve the ability to learn, to store, and remember. And this has been shown in preclinical tests where we have had young mice equivalent to age of 25 and old mice equivalent to 75, 80. And we gave them one dose of ACD856. We performed a memory test, and then we measured cognition. And when we looked at cognition after 11 days, we saw that the old mice who had not received ACD856 had more or less forgotten everything, while the young mice remembered everything. And this was also the case for the old mice who had received ACD856. And we have done a phase 1 study where we have shown that the compound ACD856 is safe and well tolerated, excellent PK profile, which shows it is suitable for once daily with a half-life of 20 hours. We also see that it goes over the blood-brain barrier, more than 37%. And we also see a central target engagement as measured by quantitative EEG, where we show that we activate areas in the brain which both are central in cognition, but also central with regard to antidepressant treatments. And now this candidate ACD856 is being prepared for phase 2.